Hello and welcome to lab number 5. Hello and welcome to lecture number 5. We're going to be looking at media and growth of microorganisms today. Again, this is Nick Cap and this is Microbiology, General Microbiology 240. And today we're going to lecture on chapter number 5 in your textbook looking at media and growth. And so some of the things that we're going to be covering are what are the requirements for microbial growth? We're going to be looking at culture media, and then we're going to be looking at how we measure microbial growth. Now, what we're looking at is kind of the growth of these microorganisms on selective media, that is, materials that are outside the body. Microorganisms growing out in the wild or microorganisms growing on the body have quite different kind of requirements and stuff. And so let me go get a pointer in here, and I'll look at this. We have a number of requirements for the growth, and these would be physical requirements, which would be things like temperature, etc., and chemical requirements, which would be the requirements for things like glucose or the food that we have. Temperature can have an effect on growth, and we've been studying this for a long time, a couple hundred years now, with microorganisms. And the psychrophiles are microorganisms that grow well at low temperatures, like in your refrigerator. The mesophiles are typically the ones that we look at that have a temperature range right about the human body, and these tend to be the pathogens that we have. The thermophiles work at kind of elevated temperature, about 70 degrees, so it's very hot water. You wouldn't be able to survive on it. And then the hypothermophiles live in kind of basically almost boiling water, and so these organisms can survive in those. And so again, if we look at this below freezing, we tend to not see microorganisms growing there. But again, we just don't know yet. We really haven't looked yet. And microorganisms growing above boiling, again, we haven't looked at it yet. And so that's some of these extreme organisms that are in there. Again, food preservation temperatures that we want to look at is that at basically the body temperature, 37 degrees Celsius, about 98 degrees, or this here is about 90 degrees, where we have the rapid growth of microorganisms. And so you can have your good microorganisms growing, or you can have your bad microorganisms growing. Again, some of these microorganisms produce toxins, so even if you kill the microorganisms, the toxins may still be present. So temperatures in this range destroy most microorganisms, and then refrigerator temperatures may allow slow growth of sporulates, again, those cyclophiles that are in there. There's no significant growth below freezing. Again, let's look at pH. Again, pH is looking at the things that are acidic. Our body pH is roughly about neutral, a little bit less than neutral. Things that work at an alkaline pH are alkanophiles, and acidophiles are things that were low at that. So lactobacillus, which is one of the organisms that we're using in our examples a lot, is something that we use. And again, we use it a lot for pickling. And so here's some yogurt, here's some sauerkraut, and all of these materials. We use acid to preserve these foods and make them a little bit better. The second thing that we can look at is some basic chemical properties of looking at osmotic pressure. And so in osmotic pressure, we're looking at, here's the plasma membrane, here's the cell wall of this, and sodium concentration of 0.85%, which is neutral. At a sodium concentration of 10%, the water is going to leak out of these. And this is what we call a hypotonic solution. An isotonic solution is basically the concentration on the outside of the cell is the same as it is on the inside of the cell. And again, that's made up of a lot of water. And things like bacon, which a lot of salt or sugar, or honey, which has a lot of sugar in it, microorganisms tend to not grow very well in there because, again, they are losing their water. So again, when we look at microorganisms, we look at requirements for growth. The chemical requirements, carbon, nitrogen, oxygen, hydrogen, sulfur, and phosphorus are all needed in relatively large amounts. These are what we call the macronutrients. The micronutrients, or trace elements, are things like iron, zinc, magnesium. And again, there's different ionic ions that we have here. We also have organic growth factors, and again, oxygen it should have been up here, but that is a factor that we have in there. Chemical requirements, again, when we look at growth inside of a tube, we tend to see, again, if there's no aeration, if there's no mixing going on in this tube, we would see the higher concentration of oxygen is near the top, medium in the bottom, and then low to no oxygen in the bottom. The microorganisms that are growing in there are using oxygen and that oxygen is going to decrease. And so again, if we use this particular test, we can inoculate these, and obligate aerobes are only going to grow on the top, obligate anaerobes are only going to grow on the bottom, facultative anaerobes are going to grow a little bit better on the top, but they will grow throughout. Aerotolerant anaerobes will grow all the way through, but again, there's no oxygen or increase, and microaerophiles will only grow in their activity. And these are what we call thioglycolate tubes. And thioglycolate incorporates, sequesters the oxygen inside there, so we tend to not see it as much. Another way to reduce the oxygen and control the oxygen is by using an air chamber here and a glove box over here. And this, we can pump out the air, we can chemically remove the oxygen in some way. In culture media, this is what we grow our bacteria on. Sterile, has no living microorganisms. An inoculation was where you introduce some microorganisms into your media. And we tend to want to do things what we call monoculture. And so, again, these microorganisms that we have in here, we have peptone, which is partially digested protein, beef extract, sodium chloride, agar, and water. Agar is 15 grams, so this solidifies it and allows us to make these plates. Here's the microorganisms kind of growing on the bottom of these tubes here. And so again, if these nutrients are right and the microorganisms can use them, they will grow inside of this media. And so in selective media, what we see is we suppress unwanted species from growing, and we encourage things that we want. And so again, here are these plates that we have. Phenyl alcohol, these are organisms that can use this, and again, we have different organisms that can grow in here. Differential medias distinguish microorganisms from kind of one another, and we will try and look at that a little bit. And here's a McConkey's plates. The lactose fermenter E. coli are bright pink, and Serratia marcescens, which does not, it looks tan in this medium. Here's non-selective medium, which all microorganisms grow, and there's two different organisms we look at in here. And here's selective medium, which only one species will grow, and we can look at that. And so what this is looking at is how do we isolate pure culture from a mixed one, is we are streaking, and we're going to do a lab on this where we will streak that plate. So we have that streak plate, and we have individual colonies, and then we take from those individual colonies and can do more. And we can separate, we can isolate and separate mixtures of bacteria. If they allow us to, some microorganisms won't grow in here. And again, here's looking at streak plates. This is how we do the streak plate. We will have a lab on this, and you will be able to do that. Growth of bacterial cultures occurs by binary fission. And then what we see is one microorganism becomes two microorganisms. Each of those two microorganisms grow and become some. And this is what we call exponential growth on here. And so 1, 2, 3, 4, in 4 generations, we have lots of bacteria. And this is 2 to the nth power. This is population at that time, the original population times the number of growth curves. And so what we tend to see is this growth. So here's where we have a logarithmic of living bacterial cells. We have this log phase where bacteria grow. They reach stationary plateau because basically cell rate of death is equal to the rate of their being created. Decline is when we have death. And basically what's going on here is we have this rate limiting step. There's something controlling the growth, something that's lacking. In this lag phase, there's no increase in the number of cells. And we will have a lab on this as well. How do we do this for a direct measurement? We do what's called a serial dilution, and we'll talk a little bit more about these serial dilutions in laboratory. We have the pour plate method of the cells, where you mix up the bacteria in some agar, and you will look on the plates. We also have the spreading plate, and we've done that in class. Most probable number is actually what we're going to be doing in our laboratory, and so we'll set this up in laboratory. In direct measurement, we can take actually a filtration unit, and we can put the bacteria through a filter, and we can count the number of bacteria in the filter. We tend to do this when there are very large volumes, very small numbers in large volumes that are involved. Another direct measurement that we can do is we can use a microscopic account, and we may try this a little bit inside of our laboratory. Another indirect method is using a turbidity looking spectrophotometer, and so when we have cultures that are growing that we know about, we can shine a beam of light through there, and you can see how much light gets through, and that will give us an idea of what's there. Next, we're going to look at microscopy and take a look at these images. And so, again, thank you very much. That was lecture number 5, looking at media. And we covered things like how to look at growth, what growth is, and we'll talk a little bit more. And so this was biology 240, and I appreciate you listening to me. Thank you very much.